So my name is Tony Lee. I'm from a company called MEDIPOST, and we work on allogenic umbilical cord blood-derived MSC pipelines and also seed cell platform using cord blood-derived MSCs. So here's forward-looking statements. So a very brief background overview of our company. We were founded in 2000 in South Korea, and that's where we are headquartered, with currently about 300 employees. We have been publicly traded on KOSDAQ, which is a NASDAQ equivalent in Korea, since 2005. And our global clinical development activities for our MSC products focus on outside Korea, that is, focusing on the US as well as Japan. So one area of our business in Korea is that we operate private cord blood bank as a so-called cash cow. We cover about 43% domestic cord blood banking market, and that turns around about $20 million US revenue top line every year. And we have been doing this since 2000. And on the other side of the business, we're doing a commercialization R&D using allogenic cord blood as a raw material, starting material, and the MSCs derived from it. And we've managed to put on the market first allogenic MSC product approved by the Korean Regulatory Agency back in 2012. This is a product called Cardistem for knee osteoarthritis. I'll show some data on that. And also that's been on the market for almost 9 years. And we own and operate our own GMP facility in Korea for manufacturing of such MSC products. There's some 3,000 square meters of flow space. And here's our product pipeline that are on clinical stage as well as commercial. At the top, we have Cardistem. This is for knee OA. In Korea, the BLA was approved in January 2012, and we are currently selling around about 4,500 to 5,000 doses every year, commercially released and sold within Korea. In Japan, we've just started phase 3 trial approved by PMDA as a single pivotal for full approval. That's started enrolling towards the end of, will start enrolling towards the end of this quarter. And the US will be beginning our phase 3 trial next year in 2022. In Malaysia, with their recently introduced guideline on cell and gene therapy products, we have filed for a BLA based on our Korean approval that was back in March this year, and we expect to see the outcome of that application for market authorization next year. In Korea, this knee OA product, Cardistem, which, as I said, is a cord blood-derived MSC product, we are indication widening that into an ankle joint OA as a single phase 3 pivotal study, and the readout will be coming out next year as well. And the next one in the pipeline is a product called Pneumostem, again, cord blood-derived MSC product. Target indication is what's called bronchopulmonary dysplasia, or BPD. This is an orphan indication for prematurely born infants, a lung developmental disorder. And we have done phase 1 and phase 2 randomized control trial with 5-year follow-up data, which I'll show you a few slides down, in Korea, as well as phase 1-2 in the US with IND for 2-year follow-up with some very encouraging results. Last on the clinical stage pipeline is Neurostem. This is for advanced stage Alzheimer's disease patients. For phase 1 completed for safety, and phase 2 has also been completed in Korea, and FDA IND has been cleared as well. So this is a layout of our GMP manufacturing facility where we manufacture our commercial product as well as clinical development product, and collectively are approximately about 3,000 square meters of floor space with, in first floor, 11 clean rooms. As you can see at the bottom of the screen, 6 clean rooms. On the second floor, we just expanded there with in-house QC. So this is a product that's been approved and marketed in Korea with Cardistem, labeled treatment of repetitive endotraumatic cartilage defect or degeneration, including degenerative osteoarthritis, and very importantly, without age limit of the patients. And this has been developed with Samsung Medical Center, and basically the product comes in 2 components, as you can see on the picture. One vial, the smaller vial, contains actually live cells. We release in ambient temperature. That's 7.5 million cells per dose. And the second bigger vial is a freeze-dry sodium hyaluronate or hyaluronic acid as an excipient. And the 2 gets mixed into hydrogel and then is surgically implanted. And this product we release not in frozen formula, but in a real ambient temperature, so it has a relatively short shelf life of 48 hours. It must be used. This is how it's administered, and very unique MOA is whereby we actually expose the cartilage defect, or basically cartilage missing bone, in OA patients, and then do a microfracture-like drilling, which is on carton number 2, to create space and also stimulate and release the bone marrow from underneath, which will contain the endogenous cells from the patient's marrow, which would contain the progenitors for osteocytes and chondrocytes that will make the bone repair and also restore cartilage. Into those holes, the cell plus hyaluronic acid, the mixture hydrogel, is manually put in, like so in panel 4, and then the rest we'll put on top. That's the hydrogel is sticky enough, basically, to stay in place without any extra membrane or glue, and then the suture is closed. So it's a relatively simple surgical procedure, and what that actually does is, this is phase 3, our phase 3 patient trial outcome. This panel shows just the microfracture alone without the cells as a control group. And you can see on the right-hand side at 1 year time point, or 48 weeks, it fails to regenerate parts of the cartilage. When we actually implant in combination with the microfracture-like drilling, marrow stimulation, and then put our cells on top, after a year you can see on the bottom right-hand side panel that regeneration of cartilage or restoration of cartilage is taking place. And this is our phase 1/2 patients, 7-year follow-up MRI image, which really shows that regenerated cartilage that we confirm visually at 1 year is actually sustained up to 7 years. You can see the blue coloring on that MRI imaging. This has been published in 2017. So recently we published the phase 3 data, which was the randomized control at 10 sites in Korea with 5-year follow-up, including no age limit on the patients with OA. And that has been published. And this is from that publication showing really after single treatment, and that image that I showed you at 1 year time point confirming the restoration of cartilage. And the question was really extending up to 5 years after single treatment and that regeneration of cartilage, really whether that will sustain the improvement in pain and function of these patients. And the answer is yes. On the left-hand side, you can see a WOMAC scale. Lower the score, the better in this case. So the blue line is CARDISTIM treated patients. They improve, and then they stay on improved condition in terms of pain and function up to 5 years. Right-hand side, IKDC, another scoring system. Higher the score, the better. So the blue line goes up. As you can see, the red line, which is just microfracture alone, cannot sustain that improvement in pain and function, which is very well documented. So at least up to 5 years on a documented study, we confirmed that the improvement in pain and function is sustained. On market, we actually see this pattern really going on. As you can see here, in Korean market status, launched in 2012 on the right-hand side graph, we're growing annually about 43%. And as I said, we are releasing about 4,500 to 5,000 doses every year now in Korea. And it's available in 600 plus clinics and hospitals. And right now, that's about 22,000 doses released, accumulated since the launch. And that equates to about 20,000 patients, because some patients with larger size defects or cartilage damage would actually require more than 1 dose of the product, up to 3 doses, depending on the size. So the number of patients treated is actually less than the number of doses released. But this month, we are celebrating 20th patient treated commercially in Korea. We are able to conduct a post-market surveillance immediately after launch with some 626 patient registry. For on-market safety, there has been no concerns there. And as I mentioned earlier, we are really focusing on the OA treatment as a therapeutic area. And this CARDISTIM product is now trialed on ankle joint OA as an indication widening. This is in partnership with the Hyundai Bio Group in Korea. And then as a next-generation pipeline in the OA area, we have developed codename SMUP-IA01. This is basically targeting an early stage, mild to moderate symptomatic OA patients as an injectable. And this really would actually complement our product, as you can see on this slide. This is a modifying OA drug, so-called. And then on the right-hand side, we show CARDISTIM. This requires surgical administration, as I've shown you, and which will result in restoration of cartilage, which is very important. But this would really be applicable for Kargren-Loris grade 3 and 4, which is really mid to late stage patients who suffer from OA. In contrast, we're developing a second-generation product, codenamed SMUP-IA. This is really to early, mild to moderate symptomatic OA patients, KNL grade 2 to 3, early part of 3, and as an injectable, a simple intraarticular injection, really to slow down the degradation and degeneration process of the cartilage degeneration so that it really slows down the progression of the disease. And we've done phase 1 first in human study in Korea with 1-year follow-up data, which is very encouraging. There's no safety concern, but the efficacy readout is also very encouraging, and we will begin the phase 2 trial early part of next year. And this is a quick MOA slide, and what we're really trying to achieve here by using these cells as an injectable into intraarticular space, and really to drive the easiness of application, a simple injection, and with high efficiency and lower cost of manufacturing. This is the platform that we've developed, and really at 3 main areas of mechanism really is really anti-inflammatory effects as soon as they're introduced, as well as in the second panel, it inhibits effects of apoptosis of chondrocytes. So it prevents the dying of the cells that should make cartilage matrix, as well as on the right-hand side, it inhibits cartilage matrix degradation by releasing factors that goes against that process. So SMOP cell is SMUP with small proprietary culture method applied to our cord blood-derived MSCs, patent protected, and really optimizing the cell characterization as well as potency of these cells through a selection, a number 1, and then hypoxic condition culture. This is all basically patent released. And we expand them in a larger bioreactor type scale for larger scaling up manufacturing, as well as cryopreservation that would actually ensure the efficacy of these cells post-thawing of these cells prior to administration. So all these technologies and patent-protected IP is applied to our SMUP cell platform, and the pipeline that we get out of that was, as I said, the early to mid stage OA patients, so mild symptomatic OA patients, and this is going into phase 2, a randomized controlled trial next year. And also, quite interestingly, this SMOP cell technology or platform was applied and been licensed out to one of our partners, LG Chem Life Sciences, a part of LG Group, whereby they have gene of interest, a single gene of interest, and they actually introduced that to our seed cell, which is SMUP cell platform, and then use that as a vehicle to generate MCB, master cell bank, for their therapeutic pipelines. And this was about $400 million deal that we closed last year, and this is basically combining our seed cell SMUP platform and then their gene of interest and then further expansion technology of theirs, and then really come out with a combination of those two into a therapeutic pipeline. So last, I would like to just briefly touch on this indication, BPD, where we are excited to go into phase 2 next year. That will be 2B in the U.S. This has been orphan drug and fast track designated by the FDA, and also orphan drug designated by EMA. So BPD is a developmental disorder. As I mentioned to you earlier, basically the prematurely born babies have a problem with their lungs because they cannot receive air because the type 2 pneumocytes to make surfactant is absent, and therefore they need to go into ventilator, and then their lungs will become dry, and inflammation will occur, and eventually will become fibrotic. So babies born with less than 1.5 kilogram birth weight are at high risk of developing BPD, and that those numbers are there. In the United States, there are about 40,000 approximately every year will be the number of babies who are born with less than 1.5 kilograms of birth weight. And that number is slightly bigger in Europe, 50,000. So there's about 30% mortality for these babies. So 1/3 of these babies will not survive. And for those 2/3 that survive, will actually be in incubators at neonatal ICU for as long as 120 days. And obviously that means a very high cost of care, but also it comes with long-term complications such as growth retardation and also both physical and mental impairments. So we developed this with Department of Pediatrics, Samsung Medical Center, and where we're actually introducing our MSCs directly to the airway of these babies as soon as they're born to really prevent the inflammation to occur, and also to bridge that gap of the period whereby the baby's lungs are not really developed fully, including the presence of type 2 pneumocytes. And what we are trying to do here, obviously in this graphic here, is that the babies are obviously born very, very young or early at 23 to 27 weeks gestational age. and very prematurely with low birth weight. And those are the babies at high risk of BPD. And the BPD assessment, the diagnosis, will happen about 36 weeks, some 10 weeks later. And so they have a very strong mortality, high mortality, about 30%. And then, as I mentioned to you, very long periods of time under positive pressure ventilation, that's basically pressurized oxygen. And what we're trying to do here is to administer pneumostim as early as we can to reduce mortality, and as well as shorten that period of time that the babies will be exposed to high pressure oxygen. And then to really see if that can impact on physical and mental growth of these babies for the rest of their lives. And this is a data from our phase 1/2 clinical study conducted in Chicago, Rush Medical Center. Here you can see the blue line. Those are the eligible but non-participants, basically the babies who are born with a high-risk group of parameters and receive standard care alone. And the green line are the babies that received our cells. And you can see the number of days that these babies stay on positive pressure ventilation or neonatal ICU is substantially reduced by some 30, 35 days. And that's a good indication because these babies can go home early, meaning that they are actually doing much better. And this is data from our phase 2 randomized control study in Korea. There's a busy table on the left-hand side. What that really shows is that upon 7 days post-administration of our cells, we see a huge movements in cytokines that are indicative of an inflammatory environment, and that is being reduced. And what that really means in 5 years of life, so we follow these babies up to 5 years, and on the top panel, which shows that number of emergency room visits for their first 5 years of their life is significantly reduced in the babies who received our treatment versus control. And in long-term 5-year data, in terms of their development in the KDSD score, these are cognitive as well as physical growth, and you can see that the babies are doing much, much better compared to those babies who received placebo control. So my last slide summary is that we're a commercial stage company in Korea with phase 3 assets, that product in both US and Japan. And we've got phase 2 program for early stage OA as an injectable, which is SMUP platform. And we have our GMP manufacturing facility in Korea that's releasing commercial as well as clinical development products. And SMUP seed cell platform has been licensed out. We welcome any opportunity for collaboration with other partners around the globe. And the pneumostim asset I've shown you the data for is a phase 2 ready, phase 2B ready asset that's on the fast track as well as orphan drug designation. So thank you. Here's my contact details. If you have any questions, I'll be happy to receive the email. Thank you so much.